Good afternoon to all the CUET PG Botany aspirants. So, here in this session, we are going to deal with complete CUET PG Botany syllabus. Fine, right? In detail under every heading, we are going to learn this clearly, like what are the concepts we are going to cover. What is the weightage of each concept? If we can take CUET PG Botany previous year papers, what will be the weightage of each concept also, we will look into it. And we will also get a crystal clear idea on how to exercise each concept, how to connect the dots and learn. Okay. First, before entering into the complete detailed concepts, we will look into the contents, what are included, what are the chapters that are included under this syllabus. After looking into the complete concepts or chapters, we will go to the segmentation properly for each concept, after which we will look into the reference books also. So, here if we see, we have syllabus for botany, first chapter phycology and microbiology, where we learn completely about algae, bacteria, viruses, and then biomolecules and cell biology, where we learn completely about cell structure, cell organelles, cell division and biomolecules, biomacromolecules in detail, their structures, functions, biosynthetic pathways. Mycology and phytopathology, here it is completely memory based, where you have to remember whole lot of examples over here. And then archegoniate. Archegoniate is the female reproductive structure in the lower divisions of animal kingdom, like what? Bryophytes. Yeah, good. Bryophytes, pteridophytes and gymnosperms. Fine, right? And then anatomy and angiosperms. Here we will learn complete anatomy, internal structures, and then sexual reproduction of flowering plants, where we will learn complete reproductive structures, angiosperms, morphology also we will learn. Okay. And then economic botany. Here we will learn complete economic importance of mostly gymnosperms and angiosperms. After which genetics, here we will be highlighting various families as well. Families, their economic importance. And then genetics. In genetics, we will look completely into the principles and molecular biology. After which plant ecology and phytogeography, plant semantics. In plant semantics, we will highlight taxonomy, systematics, especially nomenclature of the plants, and then we will move on to the taxonomic categories in detail. Okay, this and all we will learn under plant semantics. Next, under reproductive biology of angiosperms, we will elaborate only the reproductive structures of the angiosperms, like complete details of gynoecium and andrecium, their structural descriptions, cohesion, adhesions, followed by their functional descriptions, variations in different families as well. And then plant physiology, complete plant physiology from transportation to plant growth and development we have to highlight here, including biosynthetic pathways, their energetics, bioenergetics. Then plant metabolism. Under plant metabolism, we will target on all the biosynthetic reactions in nutrition and respiration, highlighting the bioenergetics and numerical applications. After which last is plant biotechnology. Here we will learn out different tissue culture techniques, highlighting agrobacterium tumefaciens models as such. Most importantly, targeted genes with molecular markers, everything we will look here. Okay. This is a brief framework of the complete chapters that we have to deal from syllabus of botany. Fine, right, ma? Now we will move on chapter by chapter. Fine, right? Now, phycology and microbiology. Here if you see phycology and microbiology, of course under algae first we will learn general characters, its structure and standard classification of algae, that is Fritsch classification. And starting up the classification of algae, the primitive algae, Cyanophyceae, okay, where the cyanobacteria is included, its general characteristics, cell structure, thallus organization. Main role of cyanobacteria is what? Nitrogen fixation, right? So, it acts as best biofertilizer. Fine, right? And these are the examples: Oscillatoria, Nostoc and Anabaena. Next, if you see structure and reproduction of algae of three different classes: Chlorophyceae, Phaeophyceae and Rhodophyceae, highlighting these examples. If we see the diversity, Algae will show structural diversity, vast amount of structural diversity, where we can see different forms of algae from unicellular, colonial, filamentous to the multicellular forms. All types of algae should be in your mind. Okay, this is completely memory based. Okay, revise it twice or thrice, you will get fine tuned. Okay, as such you have colonial algae and filamentous and multicellular plant like algae, that is Chara. These three green algae we have to highlight under chlorophyceae. Phaeophyceae, ectocarpus and kelps. Okay, branched filamentous algae. And then rhodophyceae, polysiphonia again. Fine, right? These are the examples where we have to look complete details of it. Clear with phycology, right? Now we will look on to microbiology. Microbiology, of course, we have to go through bacteria and viruses. First, bacteria, we will highlight three classes of bacteria based on their wall structures, based on cell wall, primarily based on cell wall and based on different shapes. We have different classifications of bacteria. Of course, general characteristics will be common for all bacteria because it is for prokaryotic cell. If we see classification according to the shape, we have different classifications like cocci bacteria, Vibrio, Spirillum, rod-like, bacilli as such. But based on their cell wall structure, presence or absence of cell wall, composition of cell wall, we have four different types of bacteria. Here you can see first is Archaebacteria, followed by Eubacteria, and then Actinomycetes, and then Mycoplasma. These are wall-less bacteria. Fine, right? So, basic things we have learnt under our UG level, but here we have to highlight the diseases as well, their structures and examples in more detailed way. Here if you see bacteria, their structure, nutrition and reproduction, economic importance if you see, plant bacterial diseases and their control. Okay. Angular leaf spot of cotton, bacterial blight of the rice. This too we have to look into the mechanism of action by the bacteria. And then Archaebacteria, a brief account, Actinomycetes, general features. Followed by Mycoplasma, the diseases we need to target here is little leaf of brinjal and papaya leaf curl disease. Followed by viruses, structure, replication. When you are moving into the replication of viruses, you have to target two cycles, that is lytic cycle and lysogenic cycle. Lytic and lysogenic cycle, followed by plant viral diseases and control. What are the plant viral diseases we have to highlight here? Tobacco mosaic virus, which is causing tobacco mosaic disease, rice turgo. If you want to go through, you can also go through cauliflower mosaic virus. Fine, right? These are the complete details of what? Microbiology. Any doubts here? Is this clear, right? Phycology and microbiology is clear to us, like what are the concepts we need to cover. Next, biomolecules and cell biology. Biomolecules, we will go through many different topics like highlighting biomicromolecules, biomacromolecules, primary and secondary metabolites, their biosynthetic pathways. But here when you analyze CUET papers, mostly the highlighted concepts from biomolecules is biomacromolecules, where you have to learn carbohydrates, proteins, nucleic acids, lipids and enzymes. Of course, enzymes you will learn under metabolism as well, but here these are the molecules you have to highlight. Their structures, their biological functional roles and the biosynthetic mechanisms in detail. Okay. All the types of carbohydrates, types of proteins, everything you have to learn. Fine, right? Next is cell biology. Here if you want to go through, you can also go through primary and secondary metabolites as well. Okay. Primary and secondary metabolites. Under primary metabolites, this and all will come into picture because they will have direct role on the growth and development of the organism. Secondary metabolites, whose main function is what? Defense activity in a plant. Here you will learn about alkaloids, terpenoids, phenolics and all. Most importantly, look into the biosynthetic pathways of them. Fine, right? Next is cell biology. Cell biology, first you have to target the cell structure. How you will target the cell structure of both prokaryotic, eukaryotic cell? First, And when you are going through eukaryotic cell, we have to highlight plant cell and animal cell. Plant cell wall structure, different membrane models. We are most aware of, or often we are used to, fluid mosaic model. Okay, but then we have to go through other models also, like unit membrane model, sandwich model, with the scientist discoveries, and the composition of lipids and proteins also. For each model, the composition of lipids and proteins percentage is varying, right? That also should be highlighted, because we can expect that in match the following type of questions. Next is cell membrane structure and its functions. Then you target on semi-autonomous organelles, which are double membrane bound, like mitochondria and chloroplast, followed by nucleus, which is also double membrane bound. When you are going through nucleus, please go through the chromatin structure, chromatin condensation, where you will target of what model? Nucleosome model. Followed by chromosome structure and shapes. Okay, followed by chromosome structure. Yeah, in which phase you can see the crystal clear structure of chromosome under microscope? What is the phase of cell cycle? If you know, metaphase. Okay, in which phase you can see clear shapes of the chromosome? If you are aware of, we are almost there. Anaphase. Okay, in metaphase you can see clear structure of chromosome, in anaphase shapes. Fine, right? Then go through different forms of DNA and RNA. Fine, right? And you elaborate on DNA and RNA types and its functions. Look into mitochondrial DNA, plastid DNA and plasmids. Okay, because all of them will have double stranded circular DNA. And one common nature, semi-autonomous DNA. Why are they having same property? Because mitochondria and chloroplast have evolved from a prokaryotic cell, where we learn it clear under which theory? Endosymbiotic theory. Important, endosymbiotic theory, many a times asked question. Fine, right? Yeah, exam will be in the month of March, ma, second week of March. Clear? So you have approximately 60 days of time, and tomorrow I will be discussing the complete 60-day strategy for the exam as well. Today we are highlighting the syllabus. Will that be okay? Gokula Krishna? Yeah. Now, chromosomes, as we have learnt, right? Just now we have discussed its morphology, DNA organization, euchromatin, heterochromatin, and if you are getting the complete graphical presentation, okay, in a single picture, complete chromosomes of a cell, if you can analyze it in a single paper, that is addressed as what? Karyotype. And about special chromosomes also you have to learn. These are what? Giant chromosomes. Giant chromosomes. Giant chromosomes here include Lambert's chromosomes and Polyteny chromosomes. Next is cell cycle regulation. Cell cycle regulation includes mitosis and meiosis, followed by their significance. Here please look into the numerical applications as well. Numerical applications in connection with their ploidy. Highlight microsporogenesis, megasporogenesis, okay, all these from sexual reproduction of flowering plants. Please look into all the divisions of plant kingdom, their gametophyte and sporophytic generations. Okay, what are the structures that are included in gametophytic generation, what are the structures that are included in sporophytic generation. Based on that you will get a clear idea on ploidy, and you can easily go through the numerical applications. Fine, right? In connection you will get so many questions. Followed by mycology and phytopathology. Mycology and phytopathology, what you are going to learn here? First, fungi, its general characteristics, like how Fritz classification is standard for algae. Here we are looking into Ainsworth classification. Fine, right? Structure and reproduction. The classification of fungi goes as such, Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina and Deuteromycotina. Mastigomycotina example is albigo, Zygomycotina, mucor, Ascomycetes or Ascomycotina, Saccharomyces and Penicillium, Basidiomycotina, Puccinia you have to go through, Deuteromycotina, Sarcospora. Please look at the other examples also, like for example here Zygomycotina, Rhizopus, Saccharomyces, Penicillium, Neurospora, okay. All this comes under what? Ascomycetes, okay. Puccinia, Lycoperdon, these are the examples of Basidiomycotina. So, as such look into the other examples also, but type studies of these are mandatory, okay. Next is lichens. Here we go with lichens. First is their economic importance, followed by plant pathogens, ecological economic importance, followed by plant pathogens, fungal pathogens, bacterial pathogens and viral pathogens, and how to control the disease. Okay, biotic and abiotic practices on controlling the diseases should be known. Fine, right? Next is archegoniate. As we have discussed, archegoniate is the female reproductive structure in bryophytes, pteridophytes and gymnosperms. So, under archegoniate what all we have to cover, first under bryophytes, so it is Marchantia, Anthoceras, Polytrichum. This included their structure and life cycles. Type studies are important. Next, pteridophytes, Lycopodium, Equisetum, Marsilia. Most importantly, stellar evolution and heterospory. Most of the times you got questions on stellar evolution and heterospory because they are the marked evolutionary phases under bryophytic division. Heterospory is the first condition where you have seen in this division, right? So, these are most important. You can say the hotspots of this. Fine, right? Next, gymnosperms. Gymnosperms, you see pinus neatum structure, its reproduction and economic importance. These are the concepts to be highlighted under gymnosperms. Clear till here, right? Archegoniate, mycology, phytopathology and archegoniate. Till here it is clear. Enough. Anatomy and angiosperms. Okay. Plant anatomy, here if we see, of course, if you elaborate each concept, the syllabus goes vast, but need not to scare looking into each detail. Most of the things we are already aware of. We need to just fix it and learn. Fine. Here you see plant anatomy. We have two types of tissues, okay, meristematic tissues and permanent tissues. Meristematic tissues, again we can classify them based on their origin, based on their position. Okay. So, here if you see meristems, its types based on their origin and position, followed by histological organization of shoot and root apices, and the theories related to this like histogen theory and all, we have to go through it mandatorily. We have around 4 to 5 theories as such. Next, tissues and tissue systems. We have different tissue systems for permanent tissues like simple tissues, complex tissues and special tissues. Next, we have to go through leaf ontogeny, it is the structure of the leaf, both monocot leaf and dicot leaf, followed by internal structure diversity, stomatal openings and epidermal outgrowths over the leaf. Next, stem and root. Fine, right? Vascular cambium, its formation and function. Then look into anomalous secondary growth. Okay, stem, Acheranthus, Boervia, Begonia and Dracaena. What is the type of secondary growth? Taking the examples of these you can learn. Fine, right? Because in tropical plants secondary growth varies based on the season. As such, we have different types of woods also. Here we will look into different types of woods, spring wood, autumn wood, heartwood, sapwood. We have to go through that, but here we will look into the examples as well. Fine, right? And what is the economic importance of that local timbers? This also we will go through. So, clear with the concept of anatomy first, okay, and then we will move on to angiosperms. Reproductive biology of angiosperms. Here if you see embryology, First, history and importance, that is common general note. Anther structure is important, followed by microsporogenesis, how microspores are produced from microspore mother cells. Okay, followed by male gametophyte development. Male gametophyte in angiosperms is pollen grain, right? How is it developed? Followed by ovule structure and types, followed by megasporogenesis, how megaspores are formed from megaspore mother cells, that is it, megaspores. Microspores and megaspores will be haploid, right ma? Next, female gametophyte development. What is female gametophyte in angiosperms? Female gametophyte is embryo sac. Embryo sac is the female gametophyte. Then we will go through pollen morphology, different types of pollinations, both biotic and abiotic pollinations. Biotic and abiotic pollination. Followed by pollen pistil interaction, fertilization and double fertilization, then seed structure, seed appendages, seed dispersal mechanisms, and then endosperm, its development and types. We have two types of endosperm developments. One is free nuclear endospermic development and then cellular endospermic development. These things we have to go through. Next, embryo, its development and types. We have different types of embryonic developments as well. Next, polyembryony. What will be different types of embryonic development, like monosporic? Monosporic development is most common. We will also address this as polygonum type of development. This is from single megaspore. Single functional megaspore will give rise to the embryo sac. As such, we also have bisporic and tetrasporic. Fine, right? Next is apomixis. This is special mode of reproduction, which is asexual reproduction, but this is mimicking sexual reproduction. How? Like diploid egg will be matured to diploid embryo directly. As such, without gametic fusion or fertilization, we will get diploid embryos and diploid seeds. The seeds which are produced from apomixis are addressed as what? Apomictic seeds. I am just briefing you the concepts, like what all we are going to learn. Okay. Next is economic botany. In economic botany, as we have learned, all the commercial importance from gymnosperms, specially timbers, we are also addressing this in the anomalous secondary growth of anatomy, and then angiosperms. Complete economic importance we will go through. Okay. This is the whole concept that we are going through from reproductive biology and angiosperms. Clear, right? Next. Yeah. Here about genetics and molecular biology. First, Mendelianism only we will learn. Okay. What are the Mendel's principles of inheritance? We have three principles of inheritance. One is derived, two are derived from monohybrid cross of Mendel. One is derived from dihybrid cross of Mendel. Model organism is P, right? Pisum sativum. Why Pisum sativum also we should know. Why Pisum sativum also should be known to us, because statement based questions can be expected. So, here if you see principles of inheritance, monohybrid cross, we have two laws. What are they? Law of dominance. Fine, right. Next is law of segregation. Dihybrid cross, we have what? Law of independent assortment. Fine. Next you see dominant and recessive traits. Here learning these laws followed by exceptions of Mendelian's laws of inheritance is also important. Law of segregation is universal, we know. but law of dominance has whole lot of exceptions. Like we can see incomplete dominance, codominance, multiple allelism and pleiotropy. Fine, right? Incomplete dominance, codominance, multiple allelism and pleiotropy. And then we have to learn about non-allelic interactions like epistasis. Non-allelic interactions. Fine, right? Next, lethal genes, autosomes and sex chromosomes, followed by chromosomal theory of inheritance by Sutton and Bovary, important. And experimental verification of this is also important by Thomas Hunt Morgan. Next, dominant recessive traits, polygenic inheritance, which is exception to law of independent assortment, followed by linkage recombination and chromosomal mapping or gene mapping also you can say by Alfred Stutwant. From gene mapping we will get questions, important. They either give recombination frequencies or distance between genes, and they will ask us to identify the length or recombination frequency, okay, or percentage of linkage or distance between two genes, anything can be asked. Fine, right? Next, 2-factor crosses, 3-factor crosses, interference and coincidence, followed by gene mapping numericals and sex linkage. Okay, we cannot escape disorders at the end, okay, that comes from mutation. Gene mutations you have to highlight, gene mutations, chromosomal mutations, after which you have to go through disorders, Mendelian disorders, non-Mendelian disorders or chromosomal disorders. This and all we have to go through. Fine, right? Clear till here? Any doubt anywhere, ma? Clear, right? Yeah. This is all about genetics and molecular basis of inheritance that we have to go through. Here, plant semantics. This is a very basic concept, but there are whole lot of details that we have to go through here. First you see plant information, followed by plant description, plant history, definition of plant semantics, like what language is used to describe different plants. Different plants, their local names, most importantly scientific names we have to remember, including the families. Okay, systematics and new systematics, that is modern systematics, classification and nomenclature, phylogeny, different plant families, okay, like dicot families and monocot families, and plant morphology. Fine, right? Vegetative characters and floral characters we have to go through under plant families, both vegetative and floral characters. We have to highlight both of them, followed by palynology, learning of different pollen grains, and history and evolution of different plants, that comes under plant families only. Okay, these are the details that we have to finish it under plant semantics. Plant physiology, as I told you, we have to address it from transport in plants, that is we need to learn plant water relations. Plant water relations you have to initially learn about water potential, pressure potential, solute potential or osmotic potential. Then about osmosis, absorption and transport of water, and mineral nutrition. Mineral nutrition followed by what? Macro micronutrients, most important deficiency symptoms. Quite a times asked question. Next is transpiration, stomatal structure, stomatal movement. Opening and closing of stomata mechanism is very important now. Mechanism of stomatal opening and closing, important. Next, followed by phloem transport, its mechanism, enzymes, its nomenclature, properties of enzymes, classification. Factors affecting their activity is also important, followed by photosynthesis, its pigmentation, cyclic, non-cyclic photophosphorylation, which comes under what? Light dependent reactions. Light dependent reactions. Clear, right? Next, carbon assimilation. Here we will highlight C3 pathway, that is Kelvin cycle, followed by C4 pathway and CAM pathway, and then Respiration, we will look into both aerobic and anaerobic, as glycolysis is common pathway. Learning energetics after each pathway is important. Next, Krebs cycle, oxidative phosphorylation, ETS. Fine, right? Nitrogen metabolism, its mechanism of action. Fine, right? Next is plant growth regulators, PGRs. Plant growth promoters, plant growth inhibitor, followed by ethylene and brassinosteroid. Here you can also add on jasmonic acid. Clear? These are the details of what? Plant physiology. Followed by plant metabolism. This we will learn in biomolecules only, or else in plant physiology at least, wherein we will highlight what? Metabolic pathways like carbohydrate metabolism, lipid metabolism, nitrogen metabolism. Different cycles that I have included here are important. Protein metabolism, okay. Secondary metabolism or secondary metabolites, I have told you, right? Like how terpenoids are synthesized, alkaloids, phenolics and flavonoids, their synthesis is important. Regulation of metabolism, integration of metabolic pathways. Most importantly, while you are addressing phloem transport, look into source sink relationships in a plant. What are sources in the plant body, what will be the sink? Fine, right? This is important. So, these are the complete details from what? Plant metabolism. Fine, right, ma? Next is plant biotechnology, which is our last concept to be dealt. Fine. So, here plant biotechnology, introduction to plant biotechnology is important. Here scope and applications. Historical development is again a general note, but scope and applications important. And here plant tissue cultures, what you have to learn? First you have to have a basic knowledge on totipotency and differentiation. What type of cells are totipotent, where they can give rise to different cell lines, different pattern of development. And here culture medium, MS medium consists of what composition, followed by callus, suspension, organ cultures, meristem culture, micropropagation, somatic embryogenesis and synthetic seeds. Plant cell and protoplast culture. Here protoplast isolation and culture, protoplast fusion, hybrids and its application is important. Fine. And next, genetic transformation. As I told you, Agrobacterium tumefaciens is hotspot. Definite question we can expect, Agrobacterium tumefaciens, TI plasmid, and highlight the direct gene transfer methods like gene gun and electrophoresis. Most important molecular markers. Many at times you can see PYQs on molecular markers like RFLP, RAPD, AFLP, SSR, SNP markers. Marker assisted selection is also important here. Learn about plant genomics and functional genomics. Fine. Next, transgenic plants. What are the genetically modified crops? Examples only. You need not to learn into the mechanism of each genetically modified crop. Just examples. Fine. One such is what? Golden rice. Biosafety, ethical issues. Here this is completely memory based. Okay. We can expect hardly one question like that. And then like general note on biotechnology, we have to be updated. Okay. Next, applications of plant biotechnology like agriculture, horticulture, pharmaceutical uses, environmental biotechnology. This comes under normal industrial applications. Now we will look into the reference books. Fine, right? What are the best reference books that we have to follow? These are for the students who are preparing on your own. Okay. In our 60 days planner, which we are going to do it tomorrow, the PPTs itself will give you a standard solid support. Okay. I don't say you should not go through the references, but the PPTs or the videos that we are making here will give you a strong standard support. Fine, right? And these references you can go through easily. First is for phycology and microbiology. It is standard book, Vasistha and Sinha for algae, Fritsch, the structure and development of algae. These are the books. For microbiology, you can go through Prescott, Harley and Kling. Fine. And these are the concepts which are covered in these books. Fine, right? Next, biomolecules and cell biology. Bruce Albert's molecular cell biology is important. We can also go through Cooper, okay, and Leninger, of course, that stands as a major reference books for biochemistry. Then Phycology and Phytopathology by Pandey, okay, Pandey and Trivedi. Fungal biology will give you the complete details on mycology and phytopathology. Next, Archegoniate, okay, where bryophytes, pteridophytes and gymnosperms. Standard books include Vasista, followed by Vasista pteridophytes, and then Bhatnagar and Moitra gymnosperms, and these are the contents which are included in this. Next, Anatomy and Angiosperms by Pandey, Plant Anatomy. Sprone's classification, Morphology of Angiosperms, fine, right. Economic Botany, go through Verma and Agarwal, it will give you complete examples, ma'am. Next, Genetics and Molecular Biology by Karp, Cell and Molecular Cell Biology, this you can refer for cell biology as well, okay. Campbell, I forgot to mention here, Campbell is also the standard book, you can refer both Mol Bio, Genetics, Cell Bio, okay. Plant Ecology by Odom, Fundamentals of Ecology. Semantics, you can go through Simpson, Plant Systematics. Reproductive Biology of Angiosperms, go through Bhatnagar and Bojwani, Embryology of Angiosperms. Plant Physiology, of course, Theis and Ziegler is our main book, okay, that will give you the complete details of plant physiology, okay. So, with this complete syllabus analysis, crystal clear note on each chapter, each concept, followed by reference books. Now, we have got a clear idea on CUET Botany syllabus. In our next class, we will draw out a clear schedule for each day with date and topic to cover up 90% of your syllabus in the coming 60 days before your exam with practice questions. So, do not miss these live sessions at all. This is my humble request to all the CUET PG aspirants. This will be highly useful for you, okay. So, just you can go through the sessions, recorded sessions also, but these sessions will be useful for you. So, yeah, that is all for today's session, ma. Any of you having any doubt, you can drop your messages in the comment box, I will be addressing them, and if you are having any doubt, you can approach me, okay. That is all for today's session, ma.